Well, thanks for being here. Something about me. My professional job is arm candy. My wife keeps winning all these science awards. She's a very famous scientist, so I have to go to all these award ceremonies and dress up and look pretty. So that's my real job. So today I'm going to speak to you about the problem first, and then why we've decided to go open as a solution. And so I think we come to these things and we're all very excited about the advances and all the things, but we're actually not doing so well. We haven't had new breakthrough medicines for most of the large diseases that afflict our society, and we spend $250 billion a year in the biomedical research and drug discovery systems around the world. On top of it all, medicines aren't affordable. No one can think that this is a system that's going well and that needs to be replicated. And so why is this? And so I don't think any individual is to blame. I think the system is to blame, and the system needs to change. And here's some evidence for that. So most of the problems in drug discovery, we don't understand the disease. We still don't know what causes schizophrenia, let alone treat it, right? We have some weak hints to genes that might or might not be relevant, similar for Alzheimer's, but how these affect disease processes, we don't know, right? 50 years, we still don't know. And so let's look at what I think is a problem, and I'm looking in the mirror, and it's the academic sector. And so I say we are not doing a good job at being innovative enough. So on this graph here, this is a map of publications on 500 human genes that encode protein kinases. As you know, everyone has a name, so you can go into the digital literature and how many papers are there on kinase 1, kinase 2. And so I just ranked them from most popular to least popular before the human genome was sequenced. And remember, we invested all that money in the human genome so that we could uncover new biology and go into unknown areas. Unfortunately, we didn't. If you rank these kinases in the same order as they were ranked in 1999, from popular to least popular, most of the research in 2012, and aging was telling me it's the same in 2016, are clustered on the enzymes and the proteins we studied before we even knew these existed. There is a lack of a correlation between these genes and genetic links to disease. Sequence drivers of cancer, those kinases are all over. So the unbiased evidence says we should be looking down here. The evidence from we're not curing many diseases says we should be more innovative, but we're not. And this is a fault of the system in which professors hand out money to other professors, right? We hand out money, peer review. Well, peer review by definition is consensus. By consensus is not innovative. If you want to write a grant on this enzyme down here and you just say, I really want to find out what this protein does, we don't know anything about it, it will not get funded because there's no story behind that. We can't convince people how important it is, okay? And as a result, so I've said this before, my hypothesis is, this is an indictment on the world, right? We talk about using taxpayers' funding efficiently, and we're not. And so, again, I've said this before, our first hypothesis from these data was starting from the known fact that Canadians are smarter than everybody else, and then said, well, let's look only at the subset of papers that Canadians publish in this research area. Remember, the human genome is kind of cool, right? It's the only area in all of science we can bound the problem. There are 20,000 genes, period. In no other area of science can we map activity onto a closed system to really see how are we doing, how are we distributing our resources among the problem, okay? So the Canadians, I figure, would be pushing the envelope and the world would be chasing our you-know-whats. Unfortunately, that's not true. And you slice it and dice it by any jurisdiction and you get this same result. The literature is homogeneous. The way we hand out money is homogeneous. The way we think about science is homogeneous. The way we fund science both in the VC world and in the academic world is homogeneous. And what we get is we get unproductive science. Right? We may in our little worlds think we're doing so well. Sure, I publish lots of papers in Nature, but we're having no impact. And that is an enormous problem. So how are we going to change this? First of all, we say, okay, maybe industry which has to be innovative, maybe they're pushing the envelope. And so unfortunately that's not true either. And so if you track, remember most of these, so here's the evidence that these genes are important. These are from 2008 data showing every time there's a bar, there was a knockdown that did something interesting. So many of these kinases are interesting. Most of these are for the human genome. 10 years after, the patenting is still there because we keep publishing there, right? And so, you know, if you publish a paper that old chestnut kinase, it knocked out in this mouse, has a new indication in this pancreas, and the mouse tweets and stuff. And industry, oh, that's so exciting, I'm going to put a drug discovery program on that, right? If there's no data, there's no way you can justify the investment in a drug discovery program. This is a sick system, right? You have innovation sort of in your own minds because you can publish lots of cool papers here, but there's gaps in our knowledge that are profound. And remember, the touch point is we're not understanding many of these diseases, okay? So the system is the problem. And why we decided to go open, if that's the problem, we need to be able to use our resources more effectively. And so I'll show you today that by being open, we've been able to engage industry. Over the last 13 years, our organization's got about $200 million from the pharmaceutical industry in cash and in kind. And we never file patents. It gets science done faster because you can disseminate the information quickly. You absolutely increase reproducibility because industry and academia are both watching it and it's open, right? Redundancy, obviously, and it's very interesting. You can watch patient groups starting to fund these kinds of efforts, funding coming from all sorts of sectors, and that's the end what I'll talk about. So our first hypothesis in how to shift the academic world from where we've already been studying out to the long tail was maybe if we make reagents to those proteins that nobody studies, scientists will be induced to study them. So that was the hypothesis. So we started a project in collaboration with industry where we started to make high quality research tools to proteins that had not been studied before. And the idea is that if we make them available, scientists will use them. Okay, we published a roadmap to this in 2009, open access chemical probes to see novel target discovery. And the project status is that we've collaborated with 9 pharma and dozens of chemistry groups. We've produced 35 of these molecules. Adrian will talk about one in the next seminar that we did with Boehringer Ingelheim. More than 5,000 samples of these novel molecules have been distributed without MTAs. We've estimated we've saved the world 750 lawyer years. And over the next, we have very quantifiable objectives, and that's what we're going to do over the next few years. So the impact on drug discovery, I could have chosen lots. This happens to be a good one. This is a story about a bromodomain inhibitor that opened up the area of bromodomains as drug discovery targets. It actually started with GlaxoSmithKline, who made the core discovery, and Mitsubishi contemporaneously. And GSK had run an internal program that they didn't know what to do with. They had killed it. So they decided, let's disclose it to the public through the SGC. So they told us about it. And they told us actually pretty well what kind of chemistry to do. And so we took that knowledge and we started to collaborate with Jay Bradner, who was a clinician at Harvard, who studied patients in which we thought this target was linked to that disease. So together, we made a molecule. JQ1 tells us how much of a hint GSK told us. It's not JQ 4322, right? So pharma guided us as to what molecule to make. But the novelty is we made that molecule available to the world without restriction. We gave it away. And that was a precondition of starting that experiment with Harvard. Panagis is the first author of the papers at our lab in Oxford. And this is the molecule that we gave away. GSK published their molecule at the same time. This is the molecule that they've been developing for inflammation, but they killed the project. It's a protein-protein interaction inhibitor. It inhibits a protein called a bromodomain. There's 65 in the human genome. So not only did it show that this particular target, BRD4 or the BET bromodomains, were druggable, but the whole family was druggable. And we've gone on to work a lot on that. So true to form, we gave it away. And nothing, still pretty quiet on the pharma front until 2 scientists, unrelated to us, took our molecule, dumped them on cells and in animals in AML and multiple myeloma. And that was the trigger point for industry to go, wow, This is a target, it's druggable, and it's got an indication we're interested in. Pfizer joined the partnership then. 8 other companies joined quickly thereafter. And GSK resuscitated its KILD program and carried out its first in-man in March 2012. We published this paper in December 2010. From the time the compound was disclosed in the patent literature, which was around here, 2 years and 2 months. If you contrast that, right? So chemistry, internal, make it open. People we don't even know make the key discoveries, publish it. Industry gets interested. Exactly how it's supposed to work. With Gleevec, which is the poster child and a very analogous science situation with the chromosome translocation and the target is there, it took 6 years from first in the patent literature or first in the literature to first in man. Now, not all of this, of course, is due to openness and peace and love, but I phoned Brian Drucker, who was involved in this, and he said, you know what, 2 years of this was wasted lawyer time between a pharma and the Dana-Farber. They couldn't come to an agreement. And think of that. Just think of what I said. 2 years, this was a medicine that helps people. Count how many people died, right? Because what Brian Drucker did was move to Oregon, who signed the exact same deal that Harvard refused to sign. Took it, did the proof of concept, developed a drug. It's almost tragic. It is tragic. And so, but what's happening now in this bromodomain story, there are more than 30 companies pursuing the target class. There are 15 clinical trials ongoing by 7 or 8 different companies. The most advanced are in phase 2 already. 2 biotechs have been formed and bought for $500 million or more on the target class. So this is, on one hand, it's a cool story, but on the other hand, there's a whole whack of money on one target, right? And this is kind of common in the industry. So as you guys know, this is the drug discovery pipeline. You know, we participated in this process, making the candidate molecule available so that people could do research with it. And then what's likely going to happen is that this molecule or this program is going to fail in phase 2, as most novel targets do, because we don't understand the biology. So as much as we're excited about it, as much as it's a compelling, cool story, it's likely going to fail. And all that money being spent by those biotech companies, it's probably $10 billion already in play around this target. Will be flushed down the toilet. And you know what? Since 2012, we have no published data on how that molecule is doing in man. Nobody's learning anything. And when they fail, are they going to publish all the awards of those programs? Absolutely not. So it's going to be $10 billion of no societal value and no drug. And so, you know, this is what happens. You know, as the target goes forward, some company's going to fail, but that data won't be disclosed, so all the other ones will do the same thing. If it goes to phase 2, every patient going to be exposed that those molecules are going to be harmed, not helped. It's a dumbass situation, okay? So we need to enable more targets, not to make them exciting here. We need to do in the public domain all of this to phase 2 and share all those data. That will save billions. It will bring more novel targets to POC, and drug discovery actually will be more productive and more profitable because we'll be spending our money on targets that work. So I'm going to tell you how we're planning to do this. So a hypothesis is if we run real drug discovery projects in the open, no patents, sharing all the information, it'll be more cost effective, reduce duplication, more reproducible, and we can focus on higher risk targets. So the business model, of course, that's the aim. The second thing is we need to get resources from the community that's already been spent. Hospitals have a lot of R&D money. We have a lot of R&D money. Let's align it to make medicines. So we need to get these on board, but that's a massive donation. If you think as a business person, it's a massive in-kind donation that you don't need to spend risk capital on. The model based on the return on investment model is based on market exclusivity. The finance people out there and the in-licensing people realize that if you take a drug to market, you get exclusivity with no patent. The average period's, I would say, 8 years, right? If you sum up over the world, and an average patent gives you 11.7 years post-approval, so it's less, but it's still a lot of money. And if you can get to the proof of concept or to the NDA cheaper, it's a great ROI. But you need to choose the targets and the indications strategically, and I would favor going for the highest risk targets and maybe in the smaller markets. So now I'm going to tell you how we're thinking about, creating this ecosystem that we can align to make medicines in the open. First, we've got to create an academic network. So what we did is our academic network in the chemistry side involves many universities, but we need the hospitals. So Oxford, Karolinska, Toronto, and McGill have all agreed to collaborate with us, their hospitals, and allow us to access patient cells and collaborate with them to do target discovery in genomically characterized patient cells in the open, and they've agreed not to file patents on any result that we do with them. So immediately we have a network of clinical institutions willing to play with us in the open. And of course they get to publish all the work. In fact, the Montreal Neuro has gone one step further, and they said, we're not going to file patents on anything we do inside our institution. Okay? Anything. And this is a real sort of open door to industry to collaborate. So Guy says, neuroscience translates poorly into treatments. We're doing a shitty job. It's not because we're trying. So this was in Nature. It comes down to what is the reason for our existence. It's to accelerate science, not to make money. And so this is sort of the philosophy of Guy. He's the head of the whole, it's a very storied institute, the Montreal Neurological Institute and hospital. And so it's going open, but he used shitty in Nature, but I outdid him. I did 2 shitties in Nature. So he's still got 1 shitty to go. So can you manage, though, an open network? As a business person, you think, wow, I need to control it internally so we can meet our milestones, etc. And I'm going to tell you that we've been able to pull together disparate groups and align them and do a pretty good job so far in developing molecules. So we're focusing on TB and malaria to start, not because the model only applies to neglected diseases, but because we can get the funding to do it quickly. So the targets and hits for our drug discovery projects emerge from various Gates-funded efforts. And what we do is we carry out structure-guided drug discovery from these hits and turn them into early leads that we hand off to the next stage of the development process, which is run by the TB Alliance if it's a TB project, or Medicines for Malaria Ventures if it's a malaria project. So you have the gene discovery part done, funded externally. We do the drug discovery from hit to a molecule that works in animal models, equivalently to the best-in-class drug that has good PK/PD and no really bad tox warts. I mean, it's got to be fairly clean. Okay? So we donate funding from structural biology for our organization. The foundation pronounces money for chemistry, PK/PD, and all that stuff. Chris at the SGC manages this network. And we have very clear deliverables and a very clear target product profile to meet. The team comprises, I would say, probably 50 to 100 people in various groups around the world. We have expert drug discoverers and biologists to help. The in vitro biology is done all around the world, in America, in Switzerland, Scotland. The crystallography is done in America and in our organization. And then we sometimes run the MedChem in various places, Scotland, Texas. We do some, we outsource some CROs and pharma are involved. And the idea is to start on these targets and deliver something. And so one of the projects is a TB protein called PKS13. It was validated genetically, starting from a Novartis compound, and in the public domain, they grew resistant TB and they were mutated in this protein which linked the gene to the hit. The hit killed the TB, so this was the target for the hit. The overexpression of the target also made the TB cells resistant. We saw the structures, they're soakable, and we started with a chemical starting point from Novartis. And we've advanced that starting point to a molecule that works as well as isoniazide in animal models, and we've handed this back to the Gates. This one's gone to the TB Alliance, and they're doing the lead op and clinical development. In fact, this is a pipeline. We've only started in 2013, late 2013. We have a large number of candidate projects, a large number of hit ID, and a large lead ID. We've delivered the PKS13 early lead to the TB Alliance. We delivered a LysRS novel molecule to NMV, and some of the molecules we've delivered to, in fact, show off-target effect on cryptosporidiosis, which kills 300,000 children in Africa every year, parenthetically, and you've never heard of it. So I think what the purpose of this was to show that an academic group with the right people can drive drug discovery projects to completion and quite effectively and quite cost effectively, of course, because we're begging, borrowing, stealing money from all over. So in 2016-17, what are we going to start to do now? So clearly, we didn't talk about the SGC that much, but the SGC, the organization that I run, that has operations in Toronto and Oxford and Frankfurt and North Carolina and Brazil and in Stockholm, we are going to continue to work with our pharmaceutical partners and make these chemical probes and make them available to the public domain to open up new areas and learn more about the biology of the targets. But we're also going to take some of those probes and advance them through phase 1, well, first make a clinical candidate, and phase 1 and phase 2 clinical trials in the open. And to do that, we're going to launch 3 open source drug discovery companies, and the role of the company will be to own the IND. Yeah. And then we're going to probably start with cancer and infectious disease. We believe we can go from a hit to clinical POC using less than $5 million of risk capital. It's going to cost $20, $30 million depending on the indication, but that extra money will come from foundations, will come from governments, will come from Gates Foundation, Wellcome Trust, will come from pharma, right, in order to get the molecule from here to there. But there is some kinds of parts of the process it's really hard to find public funding for, so that's how much. The science and the molecules will be disclosed to the public without restriction on use. The science will be disclosed, but the permission to take it into the clinic will be proprietary. Okay, so I think it balances public good, disclosure of the science, with maintaining a proprietary asset that you can develop and license to a developer. And the asset is the regulatory package, which is licensed to a developer like a large pharma after proof of concept, so they can take it to market. I think that's, yep. Well, how much time do I have? 2 minutes?
You got plenty of time.
Oh, cool. I got 2 slides left just for fun. So, you know, we're playing with a lot of open kind of experiments. One of which is to collaborate with patient foundations and focus some of our effort on their work. The resonance with the patient foundations has been tremendous. So each of these patient foundations has funded us to do work in the open. This one is already progressing. We're going to start to write the clinical trial protocol now for this disease, which is a dominant mutation of a kinase. These are more early stage stuff. But I think the message is that the open access or open source philosophy aggregates funding and aligns funding. And so what you do this is that, you know, every discovery we make, we share, and so thus it is, you reduce the duplication that could occur. And to take it to the extreme, Rachel in the lab has elected to make her lab notebook available in real time. So we have electronic lab notebooks, and what she does every day now is she puts it online, and all the experiments she's done, what she's planning to do. At the top she has a layperson summary for the patients who are interested in finding out what the scientists are really doing. Of course, she's a very brave young lady because it is a risk to her career by showing everybody what she's doing in real time. But I'm pretty sure the patients would be distressed to know how we really operate in the science world, which is don't tell anybody anything until publication. And so I think that hopefully what happens is we can also induce a sort of system change in how scientists do their science. Because again, commonly now, the time between a discovery and its disclosure to the public domain in the real world is 3 years, right? When the student makes the observation, to making sure it's right, to writing up the paper, to when the student goes on vacation. The student can't write, so it comes in some weird-ass language that you have to turn back into English before you submit. And then it goes to the review process, and reviewer number 3 gives you a hard time. You've got to go back and do 18 months more experiment. But think of it, right? If the observation was important, and it was important for making a new medicine, that is time wasted. And for parents of children with disease. They don't want to waste that time. So, I mean, this is an experiment. Maybe nobody reads our webs. Maybe nobody cares about our experiments. We're going to find out. I mean, I'm not going into this thinking this is going to change the world. We're treating it as an experiment in open. We're going to monitor how many people look at our site. Did it make any impact at all? Does anyone approach it for collaborations? Is it ignored? And so, again, but I'm hopeful that it'll work. And certainly, again, this is a move that we're making that is attracting attention from the patient foundations. Okay, I'm going to stop right now. Thank you very much.